Good morning, children. Today we are going to discuss about the most important microbes which are useful for the human beings we are discussing, right? In the previous class, we started the microorganisms, how beneficial and how these are useful like that we discussed. And last class we discussed about the last chapter we discussed about the microbes which causes the diseases, pathogens like that. So in this chapter, we are going to discuss about microorganisms which are very useful for the human beings. Those are we are discussing. The first one we are discussing about lab. Lab in the sense lactic acid bacteria. Lactic acid bacteria which is useful for the formation of curd, from which one milk. So this lactic acid bacteria will act on which one, casein, milk protein, digestion of milk protein casein, mainly helpful to form the curd. So lactobacillus, what we will do first, what is the procedure of the making of curd? You already know, we are doing in the houses regularly. Okay, what we will do, first we will take the milk, we'll boil it, then we can cool maximum on moderate temperature, then we will add the yesterday's curd or buttermilk, that is called lactobacillus containing inoculum. In the 40 degrees centigrade we will add. So then what will happen? Curd will be formed. So the curd contains, what is the importance of curd? Curd contains so many vitamins, and also curd contains organic acids. Curd contains which one? Vitamins and organic acids. Next important structure is yogurt. What is yogurt? Yogurt is a bacterial fermentation product. So same fermentation of milk through the lactic acid, we will get the yogurt. So streptococcus, which bacteria we will use the making of yogurt? Same, streptococcus thermophilus, lactobacillus bulgaricus. What are the microbes which are useful for the making of yogurt? Streptococcus thermophilus, lactobacillus bulgaricus. They coagulate the milk protein lactose to lactic acid. So lactose to lactic acid will be formed. So these are all the things very important in the making of yogurt preparation. So this coagulates the milk protein. The lactose to lactic acid will be formed. The flavor to yogurt. Who will give flavor? Smell we will give. There will be one more compound which will form after fermentation, that is called acetaldehyde. Acetaldehyde will give flavor to the yogurt. Next come to cheese. What is the cheese? Cheese is also, very well, we will use in the making of some sweets, some some items, food items. So it is also helpful for, it is also by lactic acid bacteria, some some useful bacteria, lactobacillus, like that we will use. So here what we will use, what is the procedure in making of cheese? One enzyme we will use. That enzyme is rennet. Very important need bit. Rennet enzyme we will use to making of the cheese. Solids are separated. Okay, after fermentation, after everything reaction, solids are separated and processed to form cheese. There will be starter bacteria. What is the making of cheese? Starter bacteria will be there. The starter bacteria are lactococcus, lactobacillus, streptococcus. Lactobacillus, streptococcus. And paneer. Next important milk product is the paneer. Paneer also same curdling milk with lemon juice and vinegar, edible acids. So what we will do, large holes in the Swiss. This is a last year they asked this neat bit. What is the bit? This is large holes in the Swiss cheese. Why? Because of the carbon dioxide formation only making the large holes. Okay, these are the things you should have to remember. Next, which bacteria we will use making of this paneer? So what is one? Propionibacterium charmoni. What do you call the bacteria name? Propionibacterium charmoni. Then dough, idli, dosha. So we are making the dough and idli, dosha or not? So which bacteria? They are also same fermentation by bacteria. The dough is there. So which bacteria we will use for making of idli and dosha? Leuconostic mesenteroids. What is that bacteria name? Leuconostic mesenteroids. Idli, leuconostic mesenteroids we will use. Bread, baker's yeast we will use for manufacturing of the bread in the bakeries. The scientific name of the baker's yeast is Saccharomyces cerevisiae. Even alcohol production also, ethyl alcohol production also, we will use which one, the Saccharomyces cerevisiae. Fermentation of glucose leads to which one? Ethyl alcohol, carbon dioxide, leavening, softness of the dough will get, and carbon dioxide C2H5, so ethyl alcohol makes the bread porous and soft. Next come to single cell proteins. What are the single cell proteins? These are the nothing but what called algae, spirulina. From algae we will get maximum number of proteins. See, less time we can produce more number of proteins, that is very equal quality of the animal proteins. So these proteins also very useful for the human consumption. So which microorganisms will give the spirulina? So here the given spirulina, the spirulina we are getting from algae. Spirulina is the algae. This is producing the single cell proteins. These proteins rich food humans and helpful for the humans and even animal feed also. Rich food for the human and animal feed also, it will be there. Next come to microbes in industrial products. What are the microbes in industrial products? Say for a making of alcohol, ethyl alcohol, beer, wine, rum, such kind of beverages. To make the beverages we will use the microorganisms. Even to making of penicillin. What is a penicillin? Antibiotic. So how to, from where we are getting the penicillin? Penicillin we are getting from which one? The antibiotic. That is, you know very well, the Penicillium notatum or Penicillium chrysogenium, and organic acids and amino acids, vitamins, biofuels, and single cell proteins, enzymes, steroids, vaccines, pharmaceutical drugs. These all we are getting from the which one? By the help of microorganisms. Okay, children. The fermenter. So if you want to produce more amount, bulk amount we want to produce, in the sense, which one we have to use? We have to use the bioreactors. Nothing but what? Big vessels. So they are called fermenters. So fermenters are useful for what? We want to we want to provide the raw material. After providing the raw material, after keeping the raw material in the fermenter, we have to add the microbes. For microbes, proper conditions we have to provide. Then they will be multiply and they will produce the product. Am I right? So what kind of conditions we have to provide the fermenters? Proper aeration, proper agitation, proper temperature, proper pH, and overflow. So proper aeration, proper agitation, proper temperature, proper pH. So pH is very important. And if suppose overflow is there, that overflow from the biomass or remove the waste from the biomass, special method also will be there in the bioreactors. Okay, children, these all are very important. So what we discussed once again, so uses of microbes in the human welfare we are discussing. In that, lactic acid bacteria importance, what? Conversion of milk to curd, and casein, the casein digestion, milk protein casein. So digestion of this casein only forms the curd. So curd, how to make? For milk, what we are adding? Lactobacillus inoculum. So millions of lactobacillus is there in that. We are adding to the, after boiling and cooling the milk. So then curd will contains the what? At which temperature we are adding? Moderate temperature. So room temperature, curd 40 degrees centigrade, less than 40 degrees centigrade we will add. So vitamins, organic acids are there in the curd, very good to the health. Same like that yogurt. What is a yogurt? Yogurt also biobacterial fermentation product of the milk. So Streptococcus thermophilus we will use, and also Lactobacillus bulgaricus we will use make the yogurt. Coagulate the milk protein. So lactose to lactic acid will be formed. So the what is the, for which one? For yogurt, who will provide the flavor to yogurt? So is due to the which one? Acetaldehyde. Very important bit, you should have to remember. Next one, cheese. Cheese is also one of the important product, milk product. This one we are getting by the help of the digestion process of the enzyme rennet. The solids separated, pressed to form. So whatever the solids are formed in the coagulation or digestion of this milk related things, then we have to separate the solid and press it to form cheese. Starter bacteria, we have to add the starter bacteria to that milk related components. What are the starter bacteria we have to use? Lactococcus, Lactobacillus, Streptococcus. Remember the names. Okay, these are all the names you should have to use. Then paneer, curdling milk with lemon juice, vinegar. So what we are doing? Curdling milk with which one? First lemon juice and vinegar, edible acids. These are. So large holes in Swiss. What is the reason for the cheese? Swiss cheese, there will be more large called holes are there. Why the large holes will be there? Because of the carbon dioxide. So Propionibacterium sharmani we will use making of the cheese. Very important need bit. and this is a neat bit, right? So Propionibacterium charboni we will use. Dow for idli making and dosha making, which bacteria we will use? Fermented bacteria, leuconostic mesenteroids. What is called? Leuconostic mesenteroids. Bread for baker's yeast, Saccharomyces cerevisiae, so which is useful for the fermentation of glucose. Okay, so Saccharomyces cerevisiae also useful for the fermentation of glucose and making ethyl alcohol, carbon dioxide. So these all the things what will happen while making of the bread. So glucose, ethyl alcohol, carbon dioxide, these all form. Then what are the reactions of ethyl alcohol, carbon dioxide? The leavening of dough and carbon dioxide in ethyl alcohol makes the bread porous and soft. So Saccharomyces cerevisiae or baker's yeast main you should remember in this point. Why? Because for making of baker's yeast, Saccharomyces cerevisiae we will use. Next come to single cell proteins, spirulina. So single cell proteins with very equal protein like animal protein, same composition, same nutrition values are there in the sense, that is called, what is called? Spirulina, a protein rich food. So it contains it is useful for the humans and animal feed. Next, microbes in the industrial, what you called, products. So what kind of by using microbes in the industries, large amount of what kind of products we will get by the useful microorganisms? Number one, beverages. Number two, antibiotics, organic acids, amino acids, vitamins. Vitamins also we will get. Biofuels. Biofuels, very well, very useful without causing any pollution. Okay, so methane like that, biogas like that, methane gas. Okay, SCP, enzymes, steroids, vaccines, RDN technology, very well. Pharmaceutical drugs, fermenter. Okay, we want to use more amount of the drugs, in the sense we will use a bioreactor, fermenter, aeration. So agitation in that fermenter to create the proper condition for the microbes. What you call facilities are there to proper condition of aeration, for agitation, for temperature, for pH, like that there will be flexibility is there in the fermenter to get our maximum product from the microorganisms in the fermenter. So overflow, suppose any excess overflow is there in the fermenter, when to remove, we can remove the waste from the biomass also will be there in the which one? Fermenter facilities. Okay children? Okay, children, so some more antibiotic production. How antibiotic production is going on, that one we are going to discuss in this process, in this concept. Antibiotic production, generally what is the use of antibiotics? They will do what they will. Antibiotics kills or retarded the growth of other disease causing microbes. That's it. One microorganism is killing another microorganism. So that is only main important. That's why we are calling antibiotic. Anti means what? Against to the. Antibiotic means what? Exact meaning? Against to the life. So which kind of mainly infections we controlled by using antibiotics? Mainly we used antibiotics to control plague, meningitis, diphtheria, syphilis, leprosy, tuberculosis. So what are the diseases? Mainly we use it to control the diseases. Antibiotics we use to which kind of diseases? Plague, meningitis, diphtheria, syphilis, leprosy, tuberculosis. So Salman Waxman first discovered streptomycin and first used term antibiotic in 1943. Who is a person? Salman Waxman first used streptomycin and first discovered the streptomycin and first used the term. Which term? Antibiotic. So Salman Waxman, important bit, in 1943. Staphylococcus bacteria. See, very very important. This is first how the Alexander Fleming doing research in his laboratory about Staphylococcus bacteria. So one green mold in his petri dishes, what he identified, used and he kept separately the petri dishes about Staphylococcus bacteria growth which have that petri plate. So when they observed after some time the growth, one particular green mold grown, the green mold where the grown that particular area there is no growth of Staphylococcus. Suppose you know very well green mold is grown, then there is the surrounding no Staphylococci could not grow. Means green mold. Something, some chemical is there in that green mold. That green mold only called, that chemical only the later they identified, Fleming identified penicillin. So it is accidental invention. Penicillin is an accidental discovery, accidental invention. Penicillin is the first antibiotic. Which one is the penicillin is the first antibiotic by which one, whom Alexander Fleming, discovered by Alexander Fleming in 1926. So which penicillin, which genus fungus from that penicillin we are getting, the Alexander Fleming got? There are two fungal genuses are there, species are there. Number one, Penicillium notatum, Penicillium chrysogenium. What are the names? Penicillium notatum, Penicillium chrysogenium. Remember these two. And they are bactericidal. They kills the bacteria and they will stops the growth of bacteria. The penicillin is called, what do you call the penicillin is called? Queen of the drugs. The penicillin is called queen of the drugs. Lateral. So later, after invention, sudden invention, no. After sudden invention, later the great scientists, who are those? Ernest Chain, Howard Florey. They developed the penicillin and they used this penicillin. Who? Ernest Chain and Howard Florey. They used it, they work, what you call, developed, still they worked on perfect penicillin. They discovered, they used and they given the perfect penicillin to the society. And these two scientists, they used this penicillin, means they asked the government to use this medicine to treat the soldiers who wounded, who got wounds in the World War II. That's why the Ernest Chain, Fleming, Ernest Chain and Florey together they got which one? Nobel Prize in the 1945. They got the Nobel Prize also. This is the most important point to remember. Next, tetracycline. What we are discussing? Tetracycline. The tetracycline is, what is tetracycline? Is also antibiotic. The tetracycline antibiotic, broad spectrum. So it controls so many, what you called, bacterial infections. Broad. So it will work very broadly. Antibiotic inhibits the microbial proteins. Whatever the proteins are forming by the microbes, transcription, translation after, they controls this tetracycline. Those proteins are controlled by tetracycline. Then chlorotetracycline from which bacteria? Streptomyces aureofaciens, Streptomycin, actinomyces. These all are fungal species and some are bacterial. Streptomyces, okay, bacterial species, and streptomycin we are getting from the actinomycetes. Okay, remember chlorotetracycline we are getting from streptomyces, so fungus. Streptomycin we are getting from actinomycetes. Like that, the fungus, from the fungus we are getting these all the, what you called, antibiotics. So streptomycetes, Gracious also very helpful for the production of, what you called, antibiotic. And these all streptomycin, chlorotetracycline, tetracycline, these all are useful to control. So which one? The gram positive and gram negative. Gram positive and gram negative bacteria. So mycobacterium, and they can control, so which kind? This bacteria, all streptomycin, they will be act against to which one? This all, this against the mycobacterium tuberculosis, TB causing bacteria, erythromycin, chloromycetin. Okay, so some more examples of what you called this antibiotics: erythromycetin, chloromycetin, griseofulvin, neomycin, kanamycin, bacitracin are best examples for the antibiotics. Okay, next come to antibiotic resistance. Overuse, what will happen? Continuous you are using antibiotics, indiscriminate usage of antibiotics, using in the sense, the bacteria will get resistance against to that antibiotic. So when you misuse the antibiotics or overuse of antibiotics, what will happen? The bacteria will be get the resistance. Then there is no effect of narrow spectrum antibiotics. Then we have to use broad spectrum antibiotics. So starting itself, we should not use broad spectrum antibiotics. So we should use narrow spectrum antibiotics later. If not working, then broad spectrum. So starting, if you use broad spectrum, they will get resistance, and further more power if you use only, then they will be die. So we should not use broad spectrum. First we have to use narrow spectrum antibiotics. And the bacteria which got resistance against to the antibiotics, that bacteria only could, what you call that bacteria is called superbug. What you call that bacteria is called superbug. Okay children, listen one more time. We are discussing about antibiotics, antibiotic production. What is the use of antibiotics? They kills or retard the growth of the other disease causing microbes. What is the meaning of antibiotic? It's against life. Which kind of diseases they controlled? Mainly plague, meningitis, diphtheria, syphilis. Diphtheria, syphilis is a sexually transmitted disease. Leprosy, white patches on the skin which lose the sense. So tuberculosis, very dangerous disease. Mycobacterium tuberculosis caused to this kind of problems we control by using antibiotics. Next, Selman Waxman. Who is a scientist? Selman Waxman. Selman Waxman. Streptomycin, first discovered streptomycin and first used the term, which one? The antibiotic in 1943. Enough Staphylococcus bacteria. By using Alexander Fleming doing research on which bacteria? Staphylococcus bacteria. Suddenly he stopped the growth of in his petri plate. In that place, which one growing? Green mold. Where the green mold growing in that particular petri plate, the Streptococcus bacteria, Staphylococcus bacteria did not grow. Means that green mold have something power. Some power is there to control the growth of bacteria. What is that power? What is that chemical? Later he done research on something. That green mold is only penicillin. Fungus. That fungus only have the power to control the Staphylococcus. Like that, a sudden discovery, unfortunate or what you called unexpected discovery identified by Alexander Fleming. Later, the Alexander Fleming first antibiotic is what? Penicillin only. Alexander Fleming in 1926. So which kind of species? Fungus, mainly Penicillium notatum, Penicillium chrysogenium are very important. From that only they first identified the which one? This bacteria, what you call antibiotic. Next important, the bactericidal. This kills the bacteria. That's why these all antibiotics are bactericidal. What's the meaning of bactericidal? Kills the antibiotics or kills the bacteria. Antibiotics kills the bacteria. That's why it's called bactericidal. The penicillin is called what you call queen of drugs. Queen of drugs. Later, Ernest Chain, Howard Florey developed this penicillin. So the Alexander Fleming, discovered. Unfortunately, later who developed that penicillin injection? Two great scientists, who Ernest Chain and another important scientist is Florey. This Chain and Florey developed the penicillin. No, that penicillin they used to treat the soldiers who got wounds in the World War II. Later, the Fleming and Chain and Florey, they got the Nobel Prize in the 1945. In the 1945, they got the Nobel Prize. This is important, children. Next, tetracycline. It's a broad spectrum antibiotic. Inhibits the microbial proteins. So, like that, chlorotetracycline we are getting from Streptomyces aureofaciens. Streptomycin we are getting actinomycetes. Okay, Streptomyces gracious. These all are very useful for the which one? To control the gram positive and gram negative bacteria. So and also this acts against the Mycobacterium tuberculosis, which causes the TB. Okay, next some more examples of antibiotics are erythromycin, chloromycetin, griseofulvin, neomycin, kanamycin, dacitracin. One more time, erythromycin, chloromycin, griseofulvin, neomycin, kanamycin, dacitracin. Examples of which are this antibiotics. So antibiotic resistance. Sometimes we misuse the antibiotics in the sense the bacteria will get resistance against to the which one? Antibiotics. Then what we have to do? So that's why we should not misuse the antibiotics and we should not overuse of antibiotics. So we should use narrow spectrum antibiotics instead of broad spectrum antibiotics. So when you do like that, what will happen? So we can control the germs. Then bacteria cannot get resistance. Okay. So we should not misuse the antibiotics. Next important, the bacteria which got resistance against to the antibiotics are called superbug. Okay, children, like this. Some more topics, some more points are there about antibiotics.